What if the meat on your plate never came from a farm? No animal raised, no animal slaughtered, no blood spilled on a factory floor. Instead, it was grown inside a laboratory, behind stainless steel walls, under cold blue lights, inside machines that look more like medical equipment than kitchen tools. Scientists are creating real meat, not from fields or feedlots, but from living cells. This is lab-grown meat, also called cultivated meat, a technology that promises to change how humanity eats and challenges everything we think we know about food. This is not science fiction. This is not a future prediction. This is real, and it's already happening. At its core, lab-grown meat is real animal meat, not made from a whole animal, but from its cells, the muscle, the fat, the protein. Under a microscope, these living cells behave exactly as they would inside a body, dividing, multiplying, following biological instructions written in their DNA. Inside sterile laboratories, scientists guide this natural process, using biotechnology originally developed for medicine, the same science once used to grow human tissue. What begins as a few microscopic cells can eventually become food. But how does that transformation happen? How do scientists turn something invisible to the naked eye into meat you can see, touch, and eat? Let's break it down, step by step. The process begins with a living animal, a chicken, a cow, or a pig. In a controlled medical setting, a veterinarian takes a tiny biopsy, no larger than a grain of rice. The animal survives. There is no slaughter, no factory floor. What's removed is a small piece of tissue, barely visible, but biologically powerful. Inside it are stem cells, special cells capable of multiplying and transforming into muscle or fat. These cells carry the same genetic instructions that build meat inside a living body. In the laboratory, they become something else entirely. They become the seeds of lab-grown meat. Back in the laboratory, the real selection begins. Scientists isolate the most powerful cells, usually muscle satellite cells, the same cells responsible for muscle growth and repair inside a living body, using centrifuges and sterile tools. Everything else is separated and discarded. What remains is a microscopic population of cells, barely visible to the naked eye, but biologically extraordinary. Under the right conditions, these cells can divide again, and again, and again. From a single biopsy, scientists can generate billions of identical cells, enough, in theory, to produce tons of meat, all from a sample smaller than a grain of rice. Cells don't grow on their own. They must be fed. Inside the laboratory, scientists place the cells into a nutrient-rich liquid known as a growth medium, a carefully engineered cocktail designed to mimic what cells would receive inside a living body. It contains amino acids, sugars, salts, vitamins, and powerful growth factors. In early experiments, this liquid was derived from fetal calf serum, a controversial animal byproduct that raised serious ethical concerns. Today, companies are racing to replace it with synthetic, animal-free alternatives, because this step doesn't just shape the science. It defines the ethics, the cost, and the future of lab-grown meat itself. Next, the cells are transferred into a bioreactor, a towering machine that looks more like a pharmaceutical factory than a kitchen. Inside, every condition is carefully controlled. The temperature matches body heat. Oxygen levels are monitored with precision, and a constant flow of nutrients keeps the cells alive. The bioreactor acts as an artificial body, giving the cells everything they need to multiply. Here, growth happens at an extraordinary pace. Cells double, again and again, forming a rapidly expanding population. In just days, what began as millions of cells becomes billions, the foundation for lab-grown meat. At small scale, this process happens inside a single machine. But to feed millions, it must grow far beyond the laboratory bench. Inside vast industrial halls, Rows of bioreactors work simultaneously, each one carefully monitored, each one growing living cells at scale. Scientists move between reactors, tracking temperature, oxygen, and nutrient flow, ensuring every batch grows consistently and safely. This is no longer an experiment. It is manufacturing, a new kind of food production, clean, controlled, and engineered with pharmaceutical precision. Here, biology meets industry, and the future of meat begins to take shape. Loose cells on their own don't resemble meat. To give them shape, scientists use scaffolds, edible frameworks made from materials like collagen, algae, or plant fibers. The cells attach to these structures and begin to align, forming muscle fibers in the same pattern seen inside a living animal. Under the microscope, those fibers grow denser and more organized over time. This step is critical. It's what turns a mass of cells into something structured, textured, and familiar. Here, biology is carefully guided, and the foundation of lab-grown meat finally takes form. Muscle alone isn't enough. To look, cook, and taste like real meat, fat must be part of the structure. In the lab, fat cells are grown separately, then carefully combined with muscle tissue. Layer by layer, the two intertwine, creating the balance that defines meat's texture, flavor, and juiciness. Under the microscope, distinct muscle fibers and fat layers emerge, mirroring what we see in traditional cuts of meat. This is where lab-grown tissue begins to cross a critical threshold, from a scientific sample to something that closely resembles food. Not an imitation, but meat built cell by cell. Muscle tissue doesn't fully develop unless it's challenged. Inside the lab, scientists recreate that process. They apply subtle electrical stimulation, encouraging the tissue to contract, much like muscles do inside a living body. This stimulation signals the fibers to grow thicker and stronger over time. As days turn into weeks, the tissue matures, gaining density, structure, and resilience. Every adjustment is carefully controlled, every parameter fine-tuned. This final stage is about refinement, guiding living tissue toward its most realistic form, before it's ready to leave the lab. When the tissue has fully matured, it's time to remove it from the laboratory environment. Scientists carefully harvest the cultivated meat, using sterile tools and controlled conditions to prevent contamination. At this stage, the texture is no longer theoretical. It can be seen. It can be touched. The harvested tissue is then prepared for processing, shaped, tested, and refined before it can resemble familiar food products. What began as a handful of cells has now become real meat, not grown on a farm, but built inside a laboratory. Before any cultivated meat reaches the public, it must pass through rigorous safety testing. Scientists examine samples under microscopes, screening for bacteria, contamination, and genetic stability. Every batch is analyzed, not just for safety, but for nutrition, protein levels, fat composition, vitamins, and minerals. The data is reviewed, recorded, and compared against strict regulatory standards. For governments and health agencies, this step is critical because this isn't just a new product. It's a new way of producing food, and trust begins in the laboratory. Across the world, the places where food is made are beginning to change. Fields and farms are slowly giving way to laboratories and stainless steel machines. For some, this represents hope, a future with less environmental damage and fewer animals raised for slaughter. For others, it raises difficult questions about nature, tradition, and trust. As people sit down to eat, few will see the process behind the food on their plate, but its origins are shifting, from pasture to petri dish, from farm to laboratory. And as this new chapter of food production unfolds, one question remains: is this the future of meat, or the beginning of something entirely new?